The sliding filament theory is one long process with many different subparts. It is all started by a stimuli. A stimuli is a detectable change in the internal or external environment. For example, for a fielder in cricket, the stimuli would be the ball. The stimuli is detected by a sensory organ. For a cricketer, the sensory organ would be the eyes. Others include the nose or the ears. The next stage of the sliding filament theory is where the sensory organ, so the cricketer's eyes, sends a message showing that it's detected a stimuli, the ball for the cricketer, down the sensory neuron. The sensory neuron acts like a postman for the message and delivers it to the brain, which is part of the central nervous system. The brain receives the stimulus and then sends an impulse towards a muscle so that it can move and react to the stimulus and catch the cricket ball. This impulse is sent down the spinal cord, and both the brain and spinal cord are part of the central nervous system. The impulse is called action potential. The impulse or action potential goes via the motor neuron as it goes down the spinal cord. The motor unit is part of the peripheral nervous system, which is located in the brain and the spinal cord. The impulse goes down the nodes of Ranvier, which are the bare bits of an axon, and not the myelin sheath, which are the fatty bits of an axon. The motor neuron carries the impulse to the neuromuscular junction. This is the gap or synapse between the motor neuron and the muscle fibers. If there is enough acetylcholine, this impulse is able to do saltatory conduction. This is where the impulse jumps over the gap. Acetylcholine. Acetylcholine is a chemical which is stored in vesicles, which are tubes containing sarcolemmas in the muscles. The acetylcholine helps the impulse or action potential to cross the synaptic cleft, which is the gap between the neurons at a nerve synapse. When the impulse or action potential gets to the receptors, it spreads across the muscle fibers. T-tubules and sarcoplasmic reticular. The impulse goes down the T-tubules to the sarcoplasmic reticular. The action potential goes to the T-tubules, goes through the T-tubules, which are extensions of the sarcolemma, and that go into the center of the skeletal and cardiac muscle cells. The T-tubules spread through the muscle fibers. The sarcoplasmic reticular is where calcium is stored. When the action potential gets to the sarcoplasmic reticular, it triggers the release of calcium. Actin binding site. When the muscle isn't contracting, troponin and tropomyosin cover the actin binding site, meaning that the myofibrils, myosin and actin can't connect. However, the calcium removes the troponin and tropomyosin that covered the actin binding site, so the myosin filaments are now able to attach to the actin, which forms a cross bridge. The role of ATP. ATP is like the coinage of energy in a usable form. It is the way energy is stored and used and how cells get energy. When ATP is broken down, energy is released. This means the myosin can pull on the actin filaments, which shorten the sarcomeres. Sarcomeres are each section of a myofibril. Myofibril is a unit of a muscle cell. When the sarcomeres slide over each other, the sarcomere shortens because the ends are pulled closer together. This happens along the whole length of each myofibril in the muscle cell. This is the sliding filament theory. The ratchet mechanism. Myosin detaches from actin so that the cross bridge is then broken when a molecule of ATP, which is like the coinage use of energy as shown in the previous slide, in a usable form, binds to the myosin head. When the ATP is broken down, The myosin head can also reattach to the actin binding site further along the actin filament and repeat the pulling motion. This repeated pulling motion of the actin over the myosin is known as the ratchet mechanism. At the end of the sliding filament theory, the muscle can contract. For example, the cricketer's bicep brachii and other muscle in his arm will move and contract to catch the cricket ball. The muscle contracts, which causes it to move. Once the impulse or action potential stops after the muscle has contracted, calcium is pumped back to the sarcoplasmic reticulum and actin goes back to its resting position, causing the muscle to lengthen and relax, and troponin and tropomyosin to again cover the actin binding site. Motor neurons. Motor neurons are made up of many things: the dendrites, nucleus, cell body, axon, and insulating myelin sheath. The dendrites collect electrical signals, whereas the cell body integrates incoming signals and generates outgoing signals to the axon. The axon then passes electrical signals to dendrites as another cell to an effector cell. The myelin sheath is a protective fatty covering of the axons rather than the nodes of Ranvier, which are the bare bits of the axon. The myelin sheath insulates axons and speeds up impulses. Motor units are also an important part of muscle contraction because it is made up of a group of a single nerve cell or a neuron that innervates a group of skeletal muscles. The neurons receive signals from the brain and stimulate all of the muscle fibers that are located in that motor unit. There are different sizes of motor units. The more muscle fibers, the bigger the motor unit. As soon as the motor unit gets a signal from the brain, all of the muscle fibers in that motor unit will contract at the same time with full force. This works in an all or none fashion. Myofibrils are rod-like structures that are extended along the whole length of each single muscle cell. There are three types of muscle contractions. One of these is concentric contraction. Concentric contraction is an example of isotonic muscle contraction. Concentric contraction is when a muscle shortens to produce tension. The two bones are pulled together by the force produced. This causes joint movement. For example, in football, a football player's bicep femoris is concentrically contracting when they pull their leg back to shoot. Another type of isotonic muscle contraction is eccentric muscle contraction. This is when a muscle lengthens to produce tension rather than shortening like concentric. Eccentric muscle contraction resists forces like gravity to control joint movement. An example of eccentric contraction is in the downward phase of a bicep curl. The bicep brachii eccentrically contracts to lower the weight whilst controlling it. Finally, a different type of muscle contraction is isometric. This is when the muscle contracts but it doesn't change length, unlike eccentric and concentric muscle contraction. An example of isometric contraction is the abdominals during when a volleyball player jumps in the air to hit the ball, because the volleyball player's abdominals isometrically contract and don't move or change length. The abdominals will create tension as they pull on the tendon attachment, which is where muscle is attached to the bone, but no movement is made. The posture of the volleyball player will be maintained by the muscles isometrically contracting because they don't move or change length.